Hello, my name is Lucas Reina Carsons, and today we'll be talking a little bit about my work entitled MBL lung tissue expression in COVID-19 postmortem biopsies compared to other pandemic viruses. The SARS-CoV-2 pandemic has killed over 4 million people and still poses various challenges to scientists and health professionals all over the world. The COVID-19 pathology isn't yet fully understood, but literature describes it as a systemic disproportionate inflammatory response associated to a prothrombotic state. Menin-binding lectins, or MBL, are a complement system molecule activator and opsonin, which means that they bind directly to pathogens in our body and start signaling for a leukocyte and white cell response. It is also responsible for a cytokine secretion once it's identified both by endothelial cells or white cells, which also leads to inflammatory response and can even crosstalk to a coagulation cascade activation. The complement system, on its turn, is an innate immune response, and it works by opsonizing antigens and signaling for leukocytes and macrophages, which molecules must be phagocyted and processed and attacked. This study aimed to measure and compare tissue expression of MBL in lung postmortem biopsies performed in patients that died from COVID-19 and H1N1 in the pandemics of 2009. The study also aimed to analyze possible MBL activation mechanisms that may take place in COVID-19 pathophysiology. We compared postmortem biopsies using lung samples from 24 COVID-19 patients, 11 H1N1 flu patients, and 11 non-pulmonary death causes that were our control group. Samples were collected according to all relevant National Ethics Committee rules. Lung samples were obtained through a mini thoracotomy, and the samples were then blocked in paraffin, and slides were prepared and stained with an anti-MBL stain. The slides were digitalized and then analyzed through ImagePro Plus software. When statistically analyzed through the student's t-test in the GraphPad Prism software, the COVID-19 group had a higher expression all around, and that expression was statistically significant. I was lucky enough during this project to learn how to use the GraphPad Prism software, and I conducted this test myself. We also proposed the following mechanisms to MBL function in COVID-19. Well, its increased expression could lead to a disproportionate complement system activation, which is a disproportionate immune response that would activate immune response over what is generally needed. MBL may also work as a coagulation cascade initiator through interactions with C5 and factor VIII. That would mean that the patient would be more prone to developing thrombi. MBL may also be a first signal recognized by the inflammasome pathway, which would mean that interleukins such as interleukin-1 beta, interleukin 18, interleukin 4, and interleukin 6 would be secreted and therefore causing a higher inflammatory response. Taken all together, our findings help understanding why COVID-19 patients are prone to suffer from immune-mediated thrombosis episodes, as well as target organ lesions and systemic inflammatory syndrome. Future studies that analyze the complement system may prove our MBL expression role in COVID-19 hypothesis. There were also some scientific production during the time that I was involved in this project, and these are them. We also got awarded first place in the International Cardiology meeting with this work. These are my references. Thank you very much.